Welcome to Apply Network Science to Maps of Science, and in the lesson of introduction we are going to see some examples of application of datasets that have been analyzed from the perspective of network science. So starting from the celebrated work of Barabashia and Albert, they study how was the degree distribution of some examples of different contexts. The three that you have on the right were the actor collaboration graph network, in which we have that the actresses are nodes, and the links or edges are two actors are connected if they are acting in the same movie. In B, what you have is the worldwide network analyzed in the year 98 by these authors jointly with Young. And what they show is that if we consider a network in which the nodes are the web pages and the hyperlinks are represented by edges, what we have is a network that also has this distribution, which is quite similar and in a totally different context. And the third one is a network analyzed from the power grid network in the States, where we have that the nodes are stations, power stations, and two power stations are connected if they are directly connected through electric wires. So here what we saw is that from very different settings they behave in a similar way, and if we represent in the x-axis the degree in logarithmic scale and in the y-axis the probability in logarithmic scale too, what we have is that all of them with different slopes behave similarly to a line and the slopes are 2.3, 2.1 and 4 respectively to these cases. So what they saw is that these networks behave like this because nodes or in these networks they are expanding continuously by the addition of new vertices, and the new vertices that we are adding they have preference to attach to a 16 vertex, and this provokes that there will be small number of nodes with a very high degree and a large number of nodes with very small degree. Apart from this work, Pastor Satorras and Vespignani analyzed how was the epidemic spreading in these type of networks, and they were interested by the spreading of a virus in the 2001, and they show how is the properties of the virus and these networks, and if it was possible to confine the virus or not, what are the problems that can be solved, and how the infrastructure should be considered in order to avoid this. Later, Albert Young and Barabasi published how was they illustrate the differences between exponential and scale field networks, and they also analyzed these when they were studying tolerance of the networks for different types of attacks. So in this case, what they saw is that in the exponential networks, nodes are usually have the same number of links, but in the scale-free networks, they are not homogeneous. Most of the nodes will have very few links, and very few nodes have a large number of links. And here we can see that in red we have nodes with very high number of links. In green we have links with just one or two, and they are in fact the first neighbors of these nodes. And later what we have is in black we have the rest of the nodes, which are at distance 2 of the red ones. So what they saw also was properties related to how was the average degree, and they saw that this was smaller when we were considering the networks with the scale-free property. Studying also the protein networks in which two nodes are representing proteins and networks, and two nodes are connected if these proteins are participating of the same process. What we have is they also find that a scale-free pattern was satisfied, and they also showed that the phenotypic consequence of a single gene deletion in Saccharomyces cerevisiae is affected by the position of the protein in the complex hierarchical of the molecular interactions. So the study of these as complex networks shows the importance of the situation in the structure of a protein and what are the effects. And in some cases, if it's very highly connected, removing this protein will directly provoke the death of the organism. Some years later, in 2007, Goa, Kushik, Childos, Vidal, and Barabashi studied the human disease network and the disease gene network. So if we saw in the middle, what we have is on the left, we have different nodes are representing illness, and on the right, we have the nodes are representing genomes. And what we have is that each illness is connected with the genes that are affected. So if we project and if we see which ill diseases are connected but through some genes, we have the graph on the left. And if we see which genes are connected by diseases, we have the graph on the right. You can see that the nodes with higher size, they're representing that have a higher degree. And the lines that are thicker, they are representing that there are more genes shared by these two illnesses. So this permits to have a visualization of which are the diseases which are closer, in the sense that they share more genes affected. And also we can see what type of genes are more strongly correlated because they are shared by different diseases. Projecting this and including more information, you have here two pictures of this same paper, in which you can see that in the part of the diseases above, what we have is that the diseases can be joined by different types. So for instance, in green, you can see that there are different types of cancer in the middle of the region, and you can also see that, for instance, deafness on the top of the graph, but it has a lot of side effects with other illness. So it's a very interesting analysis that permits you to explore what are the connections between illness, and also permits you to determine that when some medicine or some drug is affecting or is good for one illness, probably you have to explore and try to see if this can be validated or used with a different illness. In fact, with the COVID crisis, Barabashia and Los Calzos studied what were drugs that were already approved and has similar genes to the ones affected by COVID-19, because they will be these drugs candidates to be used and needed to be tested again just to approve the change of the use because of the similarity, and this is one of the options to have a quicker response. Here I also mentioned this survey in 2011, where the authors Vidal, Kushik, and Barabashi studied what are interactome networks and diseases. So they studied different types of networks at biological levels, such as transcriptional regulatory networks, virus host networks, metabolic, protein-protein, and disease networks, and they studied different organisms, and showing that the perspective for network science can be applied not only by protein interaction networks, but to a wider sense to other type of biological networks. An example where you can find lots of resources in the biological sense is using KEGG, the Kyoto Encyclopedia of Genes and Genomes, where there you can download information that later permits to reconstruct graphs and networks as the one that you have here represented with metabolic paths. And now, just to conclude, the last example is also another work of Barabashi and another authors, in which they study flavor networks. Here, this is a case which is very similar to the ones that we have of the diseases and genes. In graph A, what we have on the left are ingredients, and on the right we have flavor compounds. And what we saw is that if some ingredient is bigger, is because it has many flavor compounds. And what we also see is that we can see, if we project on the right, that which ingredients are connected with other ingredients by sharing a lot of flavors. And what we saw is that the garlic, tomato, and the olive and black pepper, what they have is the prevalence is the higher between all of them. And we also saw that, for instance, the shared compounds between white wine, parmesan, and tomato, this is quite high. And this is one of the reasons that you can combine these ingredients and the result is good for the taste. They also study how was the distribution or the number of ingredients per recipe in different countries, and they arrive more or less to a similar distribution. And they also study other properties of the ingredients from the point of view of gastronomy and food science. So this has been a little perspective of different examples of data sets that can be analyzed through networks. And last, just to finish, I will introduce GettyShow, which is an open platform that permits you to upload information that you have from data sets, where you have notes and links, and later it will permit you easily to compute many metrics that let you to compute the properties of the networks. And also it permits you to obtain very nice visualizations that in this field of network science is very relevant, since you have to present information, people has to interact, has to understand what are the properties reflected by the visual representation. So this is all for this video.